So also hello from my side. So as Christoph already introduced me, my name is Patrick and I'm working since almost already 15 years for g.tec in the Department of Software Engineering and Research. And yeah, today it's a pleasure for me to talk about eye tracking and EEG recordings. And as Christoph already said, about simultaneous eye tracking and EEG recordings. So what are we going about to hear in the next hour? I'm going to give you an overview which eye trackers we support in our interface. I'm going to talk about how the software interface looks like, what you need to prepare it, how the synchronization is done, how you actually acquire the EEG and the eye tracking, and about the post-processing. And also going to show two use case scenarios. One time it's an ERP of a visual evoked potential and the pupil reaction simultaneously recorded, and also multimodal EXG and eye tracking while a flight simulator. Yeah, so this presentation is more about the technical aspects about it. So it's not really about all the experiments that you can do about it and all the research, but it's more a technical overview. Yeah, how it is done, how you can use it, and what you can do with it. So, but why, I want to start why we're gonna do EEG and eye tracking together. So these are the use cases that I come up with. So already mentioned the multimodal recordings. So multimodal recordings try to put together as much information as you can get. So from different sensors like EEG, ECG, GSR, respiration. And in there also eye tracking plays a big role. So also studies for neuropsychiatric disorders like autism, eye tracking is an important factor, and also neuromarketing, which basically is the study of which advertisement, which products, or which other stimuli attracts you most. So yeah, let's get started with an overview of our supported devices. So in principle, we support two devices of two brands. So one brand is the Tobii Pro, and the other brand is the SR Research. For the Tobii Pro, it splits up into wearable and screen-based eye trackers. So for the wearable eye trackers, we support the classes 2 and the classes 3. And for the screen-based, we support the Fusion, the Spectrum, and the Spark. For the SR Research, we only support the screen-based eye trackers, which is in this case the iLink 1000 Plus. So what are the prerequisites that you need to have to run an eye tracking software on your PC? So you need a PC equipped with a Windows 11 operating system. You need to have MATLAB and Simulink installed. You also need to have the g.tec Suite 2024 and the HYSYS Pro license. The Microsoft .NET framework needs to be installed. And in the case if you're using the Tobii eye trackers, you need to have the Tobii Pro classes controller software or the Tobii Pro eye tracker manager. And in case of the classes, and if you want to do the synchronization of such events, then you also need the Tobii Pro lab software pre-installed. Yeah, let's continue. How does our interface look like? So the eye tracking interface that we implemented is based on MATLAB Simulink. And g.HYSYS is the product which is dealing with all kind of data acquisition, so we integrated it there. So in the g.HYSYS, there is a separate toolbox for the eye tracking, for the Tobii Pro classes, and the as our research. The interface is capable to acquire data from all the g.tec amplifiers, from high amp, the Nautilus, and the USB amp. You can also combine it with the Unicorn BCI core indirectly via LSL stream. I think it was topic of one of our earlier sessions today. And the Unicorn Hybrid Black also comes with a Simulink interface, so you could also potentially connect or combine the Unicorn Hybrid Black with the eye tracking interface. Yeah, and once you are within MATLAB Simulink and within HYSYS, you can use all the toolboxes that are already implemented in g.HYSYS for doing signal processing or feature extraction. And you can also combine it with other toolboxes, for example, with the FNIRs. So that would be a super interesting experiment combining eye tracking, FNIRs, EEG, and EXG. Yeah, now I want to go a bit more in detail about the Tobii Pro classes 2 and 3. So because this eye tracking device is super interesting, it quite seamlessly integrates with the subject. So the subject is just wearing these glasses, and this glasses has all the electronics inside to do the eye tracking. And it also comes with a live scene view camera. So you have the vision of the subject, and in parallel you have the eye tracking, so you know exactly where the gaze is pointing on the scene that you're also filming. So that's basically if you want to have a system which integrates seamlessly with the scene view and where the patient is looking at or the subject is looking at, then the Tobii Pro classes 2 and 3 is basically the device you want to have. So some specifications of the Tobii Pro classes. So it comes with a sample rate of 50 Hertz. Yeah, it's a 1-point calibration. You have the reflecting corneal model built in. It's a binocular eye tracking, and it's working on the dark pupil tracking technique. Yeah, and as I've said before, so we have the scene camera in front and the illumination there, and also the tracking devices there. And I want to show you a short video about how to set up the Tobii Pro classes. So yeah, this is the device. The device is battery driven, so we need to insert the battery first. And the class is going to store all the recordings on an internal SD card. So it also needs to be inserted. And then you need to connect the classes itself. So it comes with an HDMI cable. So you just connect it to the base station of the Tobii Pro, and you connect it to the classes. So I just forgot to say that the Tobii Pro classes is working wirelessly, so you don't have any wires going from the eye tracker to the PC, so you are completely free to move around. Just in comparison, the Tobii Pro Classes 3, so I think it's almost the same specifications as the Classes 2. It also comes with a 100 Hz sampling rate option. You see they improved a bit how the illumination is done and how the sensors are placed. So before you had these bars there, and now they kind of more seemingly integrated it to the glass or to the lenses in front of you. So if you wear the glasses, you don't even recognize that there is something built in to these lenses. Yeah, so now we are coming to the part where the GHISIS interface comes into to play. So GHISIS is, as I said before, is working on the MATLAB Simulink toolbox. The principle of the Simulink is that you have such blocks which implements functionality and then provide some information to the Simulink model. So basically, we develop this block for you, so you can, from a library, you can drag and drop it to your Simulink model, and then you can configure it, and then it will provide these information to the Simulink model for further processing. As I already said before, it provides a network connection, so the class itself, it just activates a Wi-Fi that you need to connect, and you connect with it in a password, and then you're free to connect to the eye tracker and get the eye tracking data from the classes. For the classes 2 interface, we extract all this information that you see here. So we extract the pupil center and the pupil diameter for the right and left eye. Same for the gaze direction for the right and left eye. The gaze position, which is a normalized position in the live scene view. And also the gaze position 3D. So the other measures that I've talked about before are in the 3D space. So let me also, yeah, this is the coordinate system. So the coordinate system of the eye tracker. So the reference is in the middle of the camera, and then you have the XYZ coordinates as shown here. And you will get from the Simulink interface the positions in the 3D space in millimeters, yeah, for further processing. The only difference we have is the gaze position, because it's not coming in a 3D space, it's coming in a 2D space in relation to the full live scene view video. So we already talked about the coordinate system, so I just pass by it. So for the Tobii Pro classes, to make it work, you need to initialize the internal SD card. So the Tobii Pro classes structures everything in studies and participants. So it's necessary to you to initialize this study participant folder before you can use it. So you need to insert the SD card, you need to power and connect the device, then you go to the Tobii Pro classes controller, you create the study, you create the participant, and then you start recording in the recording dashboard, and in the live scene view you're gonna see how the overlay works, and then the SD card is set up for you. And I also want to show you a video how this initialization is done of the SD card. So yeah, the initialization process is described in the user manual, so you can always go back to the GHISIS user manual and look it up. So then we're gonna connect to our Tobii Pro classes. So it doesn't ask for the password because we already entered it once. Then the Tobii Pro classes controller will be started. It's going to connect to the eye tracker, and then, as I said before, you need to go there to the study section and create this g.tec study. And within the g.tec study, you're going to add a new participant called g.tec participant. And then you start a short recording with the just created participant. then you're basically done with the SD card initialization. Then you're ready to go. Now I want to talk about how we're going to do the synchronization of the Tobii Pro classes live scene view and the EEG. So currently we just have two separate modalities. So we have the EEG and the eye tracking, but the time base is not necessarily the same. Or we don't have any information of the live scene view and the gaze in the live scene in the Simulink model. So we need to do this step in the Tobii Pro lab. And the Tobii Pro classes is providing a synchronization signal that we're gonna feed in to our amplifiers, to the USB and PI and Nautilus. And this is the signal is what we're gonna use for the synchronization. So the next slide. Yeah, presentation is hanging a bit. So this is a schematic of the Tobii Pro classes 2 and 3 device, and we see here this small outlet here is a 3.5 connector for the data synchronization. So you can add a cable there, and you can connect it to the digital input of your G-high amp, G-Nautilus, or G-USB amp amplifier. And I also want to show a short video on how you can achieve that. So you have this special jack, and you add it there to this power to this socket, and then in this case we use a G-Nautilus. We connect the USB cable to the Nautilus, and in this case we decided to use the digital input of the Nautilus device for getting the synchronization signal. Yeah, we're also going to turn on the Nautilus, and then we are done connecting the Tobii Pro classes to the G-Nautilus. So this is one way for synchronizing the Tobii Pro classes to our EEG amplifier. Another, as you've seen before, the cable for the synchronization signal is connected to the base station of the G-Nautilus, and the base station itself is connected with cable to the PC. That means that you are basically connected, or that the subject is connected to the PC and cannot freely move around. So, but I also mentioned here in this slide that you can use an analog input of the G-Nautilus, for example. So the G-Nautilus offers a device called multi-purpose. So it's a G-Nautilus multi-purpose, which comes with a set of 1.5 millimeter safety connectors, and then you can connect the synchronization signal to the analog input of the Nautilus, and then you don't have any connection between the subject and the PC. So the subject is then completely free to move around, so you can move around the whole room or even further as far as the Wi-Fi reaches of the Tobii Pro, and as far as the Nautilus can transmit the data to the base station. So that's then the only limitation that you have. And this is how a typical Simulink model looks like for acquiring EEG and eye tracking data of the Tobii Pro classes 2. So we have the Tobii Pro block in here, which just visualizes the data in these displays, and we have here our amplifier, and the amplifier and the eye tracking data gets combined here in a MUX, and then it's just visualized and saved. So that's the simplest scenario that you can get. yeah, the block itself doesn't have any configuration, so it just shows you which output it provides. But it's doing a calibration process whenever you start the Simulink model. So this is to ensure that the calibration or that the accuracy of the eye tracking is good all the time. But you can also run an external calibration. This is also what I want to show you in a short movie. So you see here the eye tracking manager, the Tobii Pro classes controller, and you see here the calibration card. So you just need to click calibrate and put your gaze. Let me start again and stop it. You just click calibrate and focus on the calibration card, and then the calibration is done from the device, and then you're ready to go. And this process is done automatically whenever you start the Simulink model. So the first thing, if you start the Simulink model, is watching the calibration card so that the device can perform the calibration. So this ensures that the eye tracking is accurate for each session. Yeah, I also want to show you how the Simulink model works and how you can get to the models. So this video shows you that you already have a Simulink model created, but as you might know, the g.tec comes with templates for all the models. So you can go to the Simulink start page and search for eye tracking, and then you're gonna find the Tobii Pro classes and all the other eye tracking models as a template for you to create the model for. So here, yeah, we just use the g.tec device and we configure the device. In this case, we also activate the digital inputs for the synchronization input of the classes. So there is no configuration needed for the Tobii Pro classes itself. We just start the Simulink model, and after initializing the model and connecting to both devices, the data is streamed into the Simulink model, and we see it in these visualization options here. So the right eye is closed in this case, and you will see that, yeah, it will be opened shortly, and we will get some data there. Yeah, so the data is visualized here in this displays, and it's also visualized in the scope here and saved to file that you can use for post-processing in GBase Analyze or in MATLAB. The Tobii Pro Classes 3 interface looks a bit different. So for that we skipped basically all the information about the eye gaze and so on, because you have everything in the Tobii Pro Lab software. So the only thing that you can configure here is if you want to calibrate the device for each start or not. So everything else basically stays the same. So you have the SD card, and everything is recorded to the SD card. And you just get here the synchronization output, the software synchronization output that you can store along with your EEG data. And the whole synchronization process is done afterwards in the Tobii Pro Lab, which I think will be, yeah, one of the next, will be shown here in the next slide. So this is now how we do the recording and how we do the synchronization process. So the first video will show how we start the recording and how it looks like when we record. So just let me show the video. So here on the left side we have the scope with the data coming in. So a nice EEG coming in and recorded by the g.tec, and the first channel here, it shows the trigger input, which is the synchronization signal from the Tobii Pro classes. So every 10 seconds we will get such a peak here. And here we have the live scene view of our eye tracking system. So let me start it over. And you see this red circle here. The red circle is actually an overlay. So this is the scene view of the subject, and the red circle is the eye gaze overlaid to the live scene view. So you in the live scene, you see exactly where your subject is looking at currently. So now it was the Nautilus, it was the jail bottle, and now the gaze moves forward to the catalog. Exactly. So now we have a data file which consists of our EEG data and of our eye tracking data, and also of the, oh, sorry, not the eye tracking data, of the synchronization input. And now we want to synchronize the EEG to those events or to those fixations of an object in the live scene view. And this is what the next video is about. So this is the Toby Pro Lab software, and we select that we gonna create the classes project. And before we extracted the SD card from the Toby Pro classes device and put it into our PC, and then we import the recorded file from the SD card. And then we take a look at the recording. So also in here we see this red circle, which indicates where the subject was focusing at. And now we can go ahead and add some new events. So we're gonna add events for each object that we have in our scene. It's the USB amp, it's the gel bottle, the g.Nautilus object, and the catalog. So, and in the Toby Pro lab software, we can jump from each fixation to another one. So we know exactly the time point when the subject looked the first time or the second time to an object. And then in this case for the Nautilus, we spotted the point. So let me jump back a bit. So right at this time point, we noticed that the subject was focusing the Nautilus object, and then we go here to the event tab in the Toby Pro Lab, and we add this g.Nautilus event. And we also gonna see it visualized here. We get a mark for the g.Nautilus in the timeline. So we just go ahead, we jump through all the fixation of our gaze. Where it hits the object, we add the event. Yeah, so we do it for all the fixation where it hits an object. And then the next part that we need to do is to export the that we want to combine now these events that we extracted from the Toby Pro Lab software and the EEG. So therefore we yeah, one step that I missed here is that we need to export these events that we just generated in the Toby Pro Lab. So therefore we go to this data export menu and we only select the event type or event information item, and we extract all the information, or then we extract the event time points to a TSV file, and this TSV file is part of the integration process that they're gonna describe now. So we're gonna open the GBS Analyze and we load the EEG data that we recorded before. And then we have basically two options. We need to let the GBS Analyze know which channel holds the synchronization input. So we can go there and set an attribute of the channel with the name Toby Sync, or we can just skip this part and start with importing the area of interest events and define the channel afterwards. So we're going to start then the integration process. The software will ask us for the TSV file that we exported from the Toby Pro Lab. Then we need to set we need to specify which events we want to integrate and which channel holds the synchronization signal in case we didn't do so far. And then the events are imported to GBS Analyze for further pre-processing. So this shows the event selection and the synchronization channel, how it is selected. And this is the final result of the integration. So in the GBS Analyze, you will get markers for all the events that you specified. So we see here that the jail bottle, the g.Nautilus, and somewhere we should also find the g.USB amp, and I couldn't find it here, but we see the catalog here. So this is the final result, and I want to show you a video how it is done. So in MATLAB, we start GBS Analyze by typing GBS Analyze to the command line. It opens the GBS Analyze software. In here, we load the EEG data that we recorded, which holds the EEG and any other signal modalities that we recorded, and the synchronization input of the Toby. So after loading, we just rescaling the signal so that we can judge if the EEG signal is good, if the signal quality was good enough. And we just visualize a longer range of our EEG. And then we go ahead and start the integration or the import of the area of interest events that we generated before. So it's this TSV file that we get from the Toby Pro Lab, and here we specify the trigger channel which holds the trigger information. In this case it was the last channel, and we select all the channels, all the events from this the file, and then the GBS Analyze imports the event time points and synchronizes it with the EEG. And we already see the three lines, the green lines, which show the Toby Sync interface event. And yeah, let me stop the video here now. So we will get a marker line at exact that time point in the video where the subject looked at the g.Nautilus system or the g.Nautilus object. So we can go ahead and GBS Analyze and start epoching our data. So we can, let me move a bit forward so that we can see the full recording. Yeah, so now we see the full recording from the beginning to the end, and we see the marker lines of the events of the live stream view. And as I said before, we can go ahead and use the epoch extraction methods of GBS Analyze to extract the epochs for g.Nautilus. And then we can extract some features of the EEG around these time points where the subject focused on the Nautilus object. Yeah, so that was it for the wearable eye tracker systems of Tobii. So I want to also introduce the Tobii Pro, the screen-based eye trackers. I already mentioned that we support the Fusion, the Spark, and the Spectrum. So this table just gives you an overview of the specifications of the different eye trackers. So the Spectrum can go up to a sampling frequency of 1200 Hertz with pretty good precision and accuracy and a good latency. Yeah, the Fusion will go up to 250 Hertz, and the Spark just can just reach 60 Hertz. And yeah, so also the prices are different for the devices. So the Spectrum is the most expensive, and the Spark is the cheaper solution, depending on the application you want to use for the eye tracking. Yeah, for the Tobii Pro, we also have a separate block for it. You can see it here in the model. So this is the Tobii Pro interface. And the only configuration that we need to do in here is to specify the sample rate we want to upsample the eye tracking data to. So normally you want to upsample it to the EEG sample rate. If you use g.tec, it's 250. If it's a high amp or USB amp, it's normally 256 or any other sample rate. So it will be from the block itself upsampled to the sample rate, and then you can combine it together, the EEG and the eye tracking of the Tobii Pro. Yeah, so for the screen-based eye trackers, we do not get as much information as we get for the Tobii Pro glasses, for example. We just, in this case, we get the gaze position and the pupil diameter. What we need to do though before is that we need to specify, or that we configure the eye tracker. So I want to show it here in this, there's the application called Eye Tracker Manager of Tobii. You can select your eye tracker there, and it gives you a position guide where you need to, where your head needs to stay. So you see here this avatar is in the correct space, and then the background is colored green. And in the eye tracker manager, you're gonna do the eye tracker setup. So you need to specify which screen you're using, the location where the eye tracker is located in relation to the screen, and also the size of your screen. This is the three minor, or the three steps that you need to do to set up the eye tracker. And then you are ready for the configuration, the calibration, and yeah, I want to show you that in a short video. So we start here, the calibration, and it wants us to focus the dots, and then it will just explode. So it will be 5 dots in total. And then the calibration is done, and we will get the calibration accuracy from the software, and we're gonna use this calibration for the eye tracker connected. There is no recalibration done in the Simulink software, so if you want to recalibrate, you need to go back to this eye tracker manager and rerun the calibration. Yeah, and this is the use case scenario that I want to show you. So this is an experiment that we run here in Heidelberg, is a visual stimulation task or experiment. And for this visual stimulation, we extracted the ERP from the EEG and also the pupil reaction from the eye tracking device. Let me describe the model a bit here below. So we have here our EEG device. In this case, it was an Nautilus system. It acquires the data and visualizes it in the scope. We do have the Tobii Pro interface, as I've shown before. It will extract the position and the diameter of the left and right eye and provide it to the Simulink model. And then we have here our paradigm block. So the paradigm block is taking care of presenting the visual stimuli to the subject. Yeah, so important to say here is that we don't have any sensors or any other detectors which are detecting the onset of the stimulation. The paradigm presenter itself is performant enough or smart enough implemented that it exactly knows the onset of the visual stimuli. So we don't need any external triggers. Software triggering is enough. So the paradigm presenter will show or present the visual stimuli and will generate a trigger output in this group ID output. And we use this group ID output to trigger the EEG data and the eye tracking data, doing an average and visualize it in our EP scope. Let me also show a video here of how the measurement was done. So let me describe what we're going to see here. On the left top, we see the scope with all the data coming in. So in this case, we had 8 EEG channels. We had the 2 pupil diameter channels of the eye tracker, and the channel 11 is showing the trigger inputs of the paradigm presenter. So the black window you see here is the paradigm presenter window, but this is just for visualization reasons. The subject was seated in front of a separate screen, so the paradigm presenter was in full screen on the external screen where the subject was looking at. And yeah, let's go ahead. So the paradigm presenter will start presenting this white pictures. And here on the right, we see the evoked potentials that we measure from the EEG and also the pupil relaxation. So we see here this red line here. The red line is the trigger onset, or when the paradigm presenter presents the white picture to the subject. So the trigger block that you can see here is configured to have some pre-trigger time and some post-trigger time, and we visualize 256 milliseconds before and a bit more than 500 milliseconds afterwards. And we see that the pupil reacts to the onset of the picture approximately 200 or 250 milliseconds afterwards. So in the literature you can find that it is around 187 to around 300 milliseconds, so this makes totally sense. And as I continue with the video, we can see whenever there is a stimuli here with the paradigm presenter presented to the subject, we see that the pupil reacts and narrows the pupil because the light intensity increased. So the pupil diameter gets dilated. So we see this deflection here. And we just extract online with the trigger block. Every time there is a stimulation onset, we extract some time before and afterwards, and then we average the reaction of the pupil and also the AP. This is what we're gonna see here. Yeah, as we go ahead and the more trials we get for our EEG, we can also see in some channels we would expect for a visual evoked potential is that we want to see an N170 response. Yeah, we don't see it that good. Maybe here it starts forming, but yeah, after such a low number of trials, you don't really see it perfectly. But if you continue with the recording and have a sufficient number of trials, we can get such a result. And we see in a set of channels, for example, here, 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 and here, we see exactly the 170 milliseconds, the 107th component in our ERP. And in the same scope, we see the pupil reaction. If you're wondering why this value here is that small, so and also here for the ERP, we often running a baseline correction, so that the baseline or that the data before the trigger is basically subtracted from our response. So the baseline is flat here for the EEG and also almost flat for our pupil reaction, and then we see basically relative to the baseline, we see the reaction of the pupil and the EEG. So we also want to use the time and describe the other manufacturer that we support. It's the SR Research iLink 1000+ device. So here we see it with a head mount, and this is the device itself. So the SR Research has one feature that is how should I say? It's the unique selling point, I guess, for the SR Research. So they can go up to a sample rate of 2000 Hertz, also in binocular measurement, and they have a pretty good accuracy and resolution, and pretty good latency constraints. How does the interface look like? So this is the architecture for the SR Research. It comes with a host PC, which is hosting the eye tracker itself, and then it's connected via a network cable to a display PC, which is taking care of the paradigm. So it's showing the text or the pictures or whatever you want to do. And we can use this network connection and connect to it as well. And then we can, once there is an established connection between the host PC and the display PC, we can connect to this established data stream and get it into the Simulink model. So here, just the same principle as before. We have our amplifier getting the EEG data, and our SR Research iLink, which is getting the position, size, and the head reference for the left and the right eye in this case. Configuration-wise, there is not that much to do for the SR Research. So you just need to specify the IP address, and you can also use the default. And here you can also specify how the EEG or the eye tracking data gets sampled to your EEG data. Last but not least, we also did recently a flight simulator demo, which was a multimodal recording. So we used the Nautilus and the g.tec to record EEG, ECG, GSR, and also eye tracking. So we used the Tobii Pro classes 2 for it. From the EEG, we just extracted some band power and we calculated the workload and the engagement measure. For the ECG, we extracted the heart rate variability, and for the eye tracking, we extracted the eye gaze fixations and the pupil size. And the subject was was operating a flight simulator, and this was the model that we were using. So here we have the data acquisition, the amplifier, and the eye tracker device. Here we extract our EEG features and the ECG features. We save everything to a file. This is advanced visualization of the features that we extract, the workload and the engagement and the RMSSD from the ECG, and just a signal validity indicator. Let me also show you that video. So this is from the Tobii Pro Lab. We also already set some markers there when he was approaching. And the interesting part is that we see when the pilot is watching the instrument, and in parallel we also extract the workload engagement and RMSSD. So I just want to put your attention to the engagement and the RMSSD values. So now we are around an engagement of 2 and an RMSSD of 40. But we see there in the background, there is already the landing pist showing up. And we see as the closer we are getting to the landing pist, the RMSSD is decreasing. So the subject is feeling some stress, and we can measure it in our ECG and the RMSSD. And also the engagement extracted from the EEG is increasing. So it shows here that we have an engagement level of 3, then before we had it to a 2. Yeah, and we see when they approach, RMSSD will stay low, the engagement will rise again. Yep, so he managed to land it properly. Yeah, and basically that was the last thing of my presentation. I just want to thank you for the attention, and if there are any questions, I'm more than happy to answer it.
Thank you very much for the nice demo. There is a question, can you use eye tracking for the blondie check? Does it make sense?
Just if you want to study at which particular region of the face the subject is looking at, so that would be the only gain and information that you would get.
So keep in mind the blondie check does something that's faster than the reaction time. So we are extracting after a couple of 100 milliseconds the information that we need for ranking the pictures. So eye tracking would be slower, and you would do it on purpose. The next question is from Chandra. Eye tracking in combination to EEG recordings. This is invasive or non-invasive? How many EEG channels?
We have just done it non-invasive. So in the demo I showed you, we have 8 non-invasive EEG channels recorded from the G.Nautilus. But there is nothing that speaks against doing it also with invasive recordings.
How many EEG channels can you use?
Yeah, the GHIAM comes with a maximum of 256 EEG channels, and yeah, that's the maximum. You can also bundle up 4 GHIAMs together, which will form 1024 channels.
But you can also do it with a unicorn with 8 channels, of course. Next one, Cora wants to know. You showed the Toby classes 2. Does it also work with Toby classes 3?
Yes. So the synchronization of the gaze events totally works with the classes 3 as well.
Jürgen wants to know, wouldn't it be better to use dynamic AOIs instead of events? I think it should be ROIs, regions of interest.
Yes. Cannot answer this question because I don't know what really this dynamic AOIs mean or what this method is about.
Just depending on the research question you want to solve. So if you want to have regions of interest that are changing and this generates trigger signals, it's okay, but you can do it, so the software is supporting it. Chandra wants to know, what's the minimum number of EEG channels to get sufficient good results?
Yeah, depends on your application. I would say starting with 8 for an ERP and starting with 16 or 32 for an MI experiment.
Santi was also coming in for the next lecture, as you could hear. Thank you very much, Patrick, for the nice demo. All the questions are answered.